Welcome to Chapter 9, How Genes and Genomes Evolve. Here are the objectives for this chapter. As always, I expect you to have these mastered by the time we take our next exam, which is coming up soon. Just like in Chapter 8, this material may be brand new, so I do not expect you to have this mastered after one viewing of the material. Make sure you take advantage of the recordings to help you hone in on these objectives. In this chapter, we are going to discuss how genes and genomes evolve. We will talk about how genetic variation is created either through sexual reproduction and point mutations, exons and gene transfers. We will wrap up this chapter by examining the human genome. All of these concepts should be building on what we have been talking about for the previous 3 chapters. For this section, we will be discussing some of the ways that genetic variation is generated. Take any random group of people, or even your closest relatives, and list out all of the visible genetic characteristics. Unless you have 2 identical twins in the pool, no 2 people are alike. Have you ever stopped to wonder how that could be? This variation doesn't just occur in humans. Within almost any population of organisms, you will notice genetic variation. Have you ever stopped to wonder where this variation comes from? In this chapter, we will discuss how this variation arises. We will then discuss how this variation actually causes evolution and how the tree of life developed from this evolution. Hopefully you remember from your earlier biology classes that evolution requires 2 things. The first of these is selective pressure, meaning something is pushing populations of organisms to change. This can be a change in temperature, loss of prey, or a variety of other factors. The second component is the ability to change through genetic variation. Just how does this genetic variation arise? It doesn't just magically appear in a generation. Instead, it is through a series of changes to the existing genome that helps introduce these variations over millions of generations. These changes in the genome come about in 5 ways. These are mutation within a gene, mutation within the regulatory DNA of a gene, gene duplication, exon shuffling, and horizontal gene transfer. In this section of the lecture, we are going to talk about each of these types of genetic changes. Before we get looking at these types of genetic variation, let's talk about how these variations come to impact an entire species over many generations. Remember, we have 2 types of cells in our body: somatic cells and germ cells. The somatic cells in our body are the diploid cells containing 48 chromosomes, and they make up most of our body. In contrast, germ cells are our sex cells. These cells contain 23 chromosomes and are eggs in females and sperm in males. Only genetic changes that occur in our germ cells will be passed on to the next generation. Let's take a look at this in terms of cancers. We have identified only a few types of hereditary cancers. These cancers arise from specific genes that are passed on from parents to offspring in their germ cells. In contrast, most of the other cancers arise from a mixture of environmental exposure and genetic predisposition. These mutations occur in our somatic cells and are not passed on to the next generation. Sexual reproduction creates a variation through shuffling combinations of genes. Asexual reproduction allows for variation to occur through genetic recombination. With this in mind, let's talk about how genetic variation arises. Remember, as we work through this chapter, all of these variations and changes we are discussing can occur in somatic cells, but our focus will be in the germ cells because those changes can be passed on to the population. Let's talk about point mutations first. We have discussed these point mutations before, such as in Chapter 4 when we talked about how the disease sickle cell has arisen, in Chapter 6 when we talked about the fidelity of the genome, and again in Chapter 7 when we discussed the impact of nucleotide substitution and the wobble effect of tRNA anticodons. Point mutations are where a single nucleic acid substitution is made within a gene. A point mutation can have no impact on the cell, creating what is known as a neutral or silent mutation. These will often occur in a wobble spot or don't create a dramatic shift in the proteins within the gene product. At the other end of the spectrum are mutations such as sickle cell, or potentially a product that is so misshapen that it will be destroyed immediately by the cell before it can do any damage. These mutations are always occurring within the cell populations. We can see an example of how quickly these point mutations can arise on this slide. Remember, the error rate in our cells is 1 in every 10 to the 9 base pairs copied. In this example, we have taken a single E. coli cell that cannot grow without histidine because it cannot create this amino acid on its own. We call it histidine negative or His negative. Wild type or non-mutated E. coli have a metabolic pathway to make histidine. The His negative cell is put into a rich media and allowed to replicate. When we sample the subsequent generations, we find that some of the cells have mutated to be able to produce their own histidine. Point mutations can also occur in the regulatory sequences in the cell. Remember those junk DNA sequences we discussed? A point mutation in this region is hard for us to find, and even harder for us to determine the ramifications of this mutation. One example of this silencing of the gene that produces the receptors on the red blood cell that prevents the Plasmodium parasite from binding and infecting the host with malaria. This is another defense mechanism to malaria that we have evolved in addition to the sickle cell mutation we just discussed. Another example of this is lactose intolerance. It is very abnormal for adult mammals to consume milk, and adults stop producing lactase after infancy. However, as we humans began to domesticate animals and drink their milk as a part of our diet, adults who had the rare ability to continue to produce lactase did better than those who had the usual regulation of gene expression. Given the improved nutrition, those who could drink the milk were then much healthier and much more likely to reproduce, further propagating the mutated regulatory gene and creating the spread that you see on this map. In countries where cows or other dairy animals were not raised, lactose intolerance in adulthood stayed the norm. Although we can see where migration has changed the balance of which gene is expressed. This goes back to our earlier discussion of what drives evolution. As humans, we have the genetic variation for how long we produce the enzyme lactase, and by domesticating animals, we made a selective pressure that encouraged the success of people who can produce lactase into adulthood. The next way that we develop genetic variation is through duplication. We have two main types of duplication: gene duplication and whole genome duplication. In gene duplication, at some point, the entire sequence of the gene is duplicated. We believe that these duplications occur due to errors made during homologous recombination. This essentially puts 2 copies of the gene on one chromosome and 0 copies of the gene on the other chromosome. You can see in this picture the mechanism. It is believed that when these errors are made, the second copy of the gene can be modified or mutated, creating a new special purpose within the cell. Let's take a look at an example of this. This example is the globulin gene family. The globulin protein, as you should be aware of by now because we've beaten it into you, is responsible for carrying oxygen into cells. In primitive animals, only the alpha chain is needed to meet the oxygen demands of the organism. However, as you can see, around 50 million years ago, this single gene became duplicated and split into alpha and beta chains. This duplication creates a very efficient 4 subunit protein that carries oxygen in larger animals. This leads us to the question, which came first, larger animals or the 4 subunit globulin molecule? Larger animals certainly couldn't have existed very well without these larger proteins, but without the need, what selective pressures could have helped these gene sequences to continue to exist within the genome? We have also been able to determine that around 20 million years ago, the beta subunit gene underwent a second duplication, creating a special fetal globulin protein that creates better oxygen delivery to the fetus. The selective pressure for this duplication is easy to see because a fetus that is able to deliver more oxygen to its tissues is much more likely to survive to birth and then be more likely to have properly developed tissues to help it survive into adulthood. Not only can genes be duplicated, but entire genomes can be duplicated. The evidence for this shows that early in vertebrate evolution, there were two duplications that occurred quickly one after the other. This then produced 4 copies of every gene that then evolved into their own unique genes to meet the needs of each organism. We can see more recent examples of this duplication in frogs. This duplication creates a huge size difference in the species. Humans believe that these duplications are results of errors during cell division, specifically meiosis. While these duplications have been shown to be an effective way to alter a large amount of genetic material in a species, in humans we have not observed that any mass duplications are beneficial. On a smaller scale, the addition of one more chromosome 21 can result in someone with Down syndrome, and the extra chromosome copy of each chromosome of the other chromosomes have shown repeatedly to be incompatible with life. So once again, these changes can arise, but only if they are successful at creating a survival advantage over the rest of the population, or at a minimum no harmful disadvantage, will these duplications be perpetrated. The next way genetic variation is created is through exons. We have 2 ways that we have seen exons participate in genetic variation. The first of these is exon duplication, and the second is exon shuffling. Let's look at exon duplication first, and then we will evaluate exon shuffling. Remember from Chapter 7 that exons are the part of the gene that codes for the actual product. If this exon is duplicated, the gene product may end up with one more protein subunit than in previous generations. This arises from the same process as gene duplication, homologous recombination, except in this case, instead of an entire gene being duplicated, only the exon and its accompanying introns may end up in the chromosome sets, leaving another pair without that exon. You can see how that plays out on the graphic on this slide. The same process that can result in exon duplication can also result in exon shuffling, when an exon is accidentally migrated to an entirely different gene. The product of that exon will then be incorporated into that gene's product. This can create quite an array of proteins from single domain additions, as you can see from the examples on this slide. It has been suggested that all proteins have arisen from exon duplication and exon shuffling. This could explain why we see such similar motifs throughout the proteins in our cells, regardless of their purpose. When we last talked about mobile genetic elements, we discussed how they can turn off genes resulting in cases of hemophilia that some historians argued started World War I. However, these elements can also create useful genetic variation. These elements can not only silence genes like we discussed, but they can also change the way the gene is regulated, changing the way very large portions of a multicellular organism is arranged. They can also result in exon or entire genome rearrangements through homologous recombination. As you can see on this slide, how these elements could cause an exon rearrangement. Below is an example of how an element can dramatically shift the body plan of an organism. Depending upon the environment that this fly is living in, the shift in appearance may prove beneficial and may shift the genetics of the entire population through improved breeding success. Although personally, I can't imagine where having legs instead of antennae would be helpful. Lastly, let's talk about horizontal gene transfer. This is a process that mainly occurs in prokaryotes, but it has been documented in eukaryotes. In horizontal gene transfer, an individual will share a genetic will share genetic information with another organism. This would be like me sharing a copy of one of my genes with you. This is an easy way to spread genetic information to the current population. Bacteria use this process to spread genes for resistance to antibiotics. Well, that's it for this topic. We'll pick up topic 2 in part 2.